In this lecture, we continue exploring the early development of mammalian embryos. We've looked at the process of gastrulation in mouse and human embryos. Recall that this process involves the movement of cells that will give rise to the mesoderm and endoderm through a structure known as the primitive streak. The primitive streak is located at the future posterior of the embryo. And in this lecture, we'll focus on how the anterior, posterior, and left-right axis of the mammalian embryo are specified. Whereas amphibians have a single signaling center, the organizer, in mammals there appear to be two signaling centers that have overlapping functions. One of these is the node, which is equivalent to the portion of the amphibian organizer that's located at the trunk region. The other is the anterior visceral endoderm, or AVE. The AVE is responsible for positioning the primitive streak, the site that marks the start of gastrulation. On the other hand, the node induces the nervous system and is important for patterning the anterior-posterior axis. As with amphibian embryos, the inhibition of BMPs and of the Wnt signaling pathway is needed for the formation of anterior structures. The trophectoderm that surrounds the epiblast secretes BMP4, and that trophectoderm is labeled here as extra-embryonic ectoderm. BMP4 causes the epiblast cells to express Wnt3 and nodal. And this Wnt3 and nodal activate transcription factors that are involved in the specification of the mesoderm. However, the anterior visceral endoderm secretes inhibitors to Wnt3 and nodal in the anterior region of the embryo. So the secretion of Wnt and nodal inhibitors by the AVE prevents the primitive streak from forming at the anterior region of the embryo. The primitive streak instead forms in the posterior region, which expresses nodal, and, as I mentioned, is inhibited from forming in the anterior region as a result of the presence of nodal inhibitors. But this pattern of nodal expression and the roles of nodal change as development progresses. The node, which eventually gives rise to the notochord, secretes BMP inhibitors like chordin and noggin. As in the amphibian embryo, the inhibition of BMPs is critical for the formation of the nervous system. And together, the inhibition of Wnt and BMP signaling promotes the formation of anterior neural structures. But how does the AVE form? Well, the AVE, which essentially determines the anterior-posterior axis by helping to position the primitive streak, is formed as a result of the constraints that are placed by the growth of the embryo inside of the uterus. So mechanical stress plays an essential role in the specification of the anterior-posterior axis in mammals. Initially, when the embryo is small, it grows in all directions. As the embryo grows in size, its growth becomes restricted by the walls of the uterus, so the embryo grows downward. As the embryo grows downward, it breaks through the extracellular matrix, and this breach in the ECM leads to changes in gene expression in the cells that are located at the site of the breach. This change in gene expression induces the migration of these cells and their specification as anterior visceral endoderm. And I should point out that this figure is a little confusing. The embryo is still located within the visceral endoderm. So these cells that are at the most distal location are the ones that are going to migrate anteriorly and give rise to the anterior visceral endoderm. As development progresses, morphogen gradients are established which are important for the patterning of the organism. Similar to what happens in amphibians, in mammals there are gradients of Wnt's, BMPs, and FGFs, with these factors being highest at the posterior and lowest at the anterior. In the anterior, BMP and Wnt inhibitors are secreted, which allow the anterior structures of the brain and the face to form. Other factors that are important for patterning the anterior-posterior axis are FGF8 and retinoic acid. The expression of these two factors is also higher in the posterior regions of the embryo compared to the anterior regions. The FGF8 gradient is formed by the graded decay of its mRNA. This happens because the transcription of FGF8 is restricted to the growing posterior tip of the embryo, and the FGF8 mRNA is progressively degraded in the newly formed tissues. This results in the formation of an FGF8 mRNA gradient and thus an FGF8 protein gradient, with the levels being highest at the posterior. In the case of retinoic acid, the gradient is established by the presence of enzymes that synthesize retinoic acid in the posterior regions of the embryo and enzymes that degrade it in the more anterior regions. These different morphogen gradients pattern the embryo, and in fact, the patterning of the posterior region of the embryo beginning at the hindbrain is related to the activation of different Hox genes as a result of the gradient of retinoic acid, Wnt3A, and FGF8.